Okay, shall we continue?
Yes, ma'am.
Okay. Regarding the regulation of pyruvate dehydrogenase, the products of the reaction, the pyruvate dehydrogenase reaction, the pyruvate will be converted to acetyl CoA, and in that one NAD will be converted to NADH. So in other ways, we can tell acetyl CoA is formed and NADH is formed. So these are the products. So when the products are present in excess, that will inhibit the reaction. The enzyme will be pyruvate dehydrogenase complex will be NADH and acetyl CoA. So pyruvate to acetyl CoA. Here you can see NADH. When NADH is present in excess, that will inhibit the enzyme. Likewise, the main product acetyl CoA, when it is present in excess, it will inhibit the pyruvate dehydrogenase. Okay. And like I've told about some enzymes, some of the enzyme will be activated in dephosphorylated form, and some will be activated in phosphorylated form. What I've told about the case of enzymes of glycolysis, active in which form? Dephosphorylated form. The same is true here also. Pyruvate dehydrogenase, the enzyme can occur in two forms. The A form, A for active. Active means it is dephosphorylated enzyme. And there is a B form which is inactive. B form means phosphorylated form. So, what all things which will convert the enzyme pyruvate dehydrogenase to dephosphorylated form will activate the reaction. Which are the things which will convert the pyruvate dehydrogenase to dephosphorylated form is that the ions like magnesium, calcium, insulin, all will convert the pyruvate dehydrogenase to A form or the active dephosphorylated form. And you can see, so this PDH can occur as A state or B state. The factors which will convert the active PDH to inactive PDH is that one is end product, one is acetyl CoA. When the ratio of acetyl CoA to CoA is high, which means the product is high, and the NADH to NAD ratio, which means when NADH is high, and when the energy status of the cell is high, all three will favor the conversion of the active PDH to inactive PDH or the phosphorylated form. Okay, and you can see here the magnesium and calcium will activate the enzyme. So here the reverse will happen. Magnesium and calcium will inactivate enzyme and decreases the rate of pyruvate dehydrogenase reaction. Is it clear for you? Yes or no?
Yes, ma'am.
Okay. So phosphorylation, the same thing is written here. Phosphorylation by a kinase, it will decreases the activity. The kinase is activated by end products, which are the NADH, ATP, and acetyl CoA. What happens in free fasting state is that more free fatty acid will be formed, and that free fatty acid on degradation, it will produce acetyl CoA. So we have plenty of acetyl CoA that will spare for the carbohydrates. In adipose tissue, this much is not needed for you. At least you study about the regulatory enzyme, which all form is active, which all inactive, what things will convert the active PDH to inactive, and the reverse. Okay. What about the role of thymine. Thymine, you know, the thymine active form is a coenzyme in this reaction, thymine pyrophosphate. So thymine pyrophosphate, when it is deficient, it will inhibit the pyruvate dehydrogenase reaction because it requires that in the initial step itself, PPP is needed. So when in alcoholic, there will be a thymine deficiency that will inhibit the pyruvate dehydrogenase reaction. And when there is excess of pyruvate, that will be accumulated and it, as it cannot move on to the acetyl CoA due to a defect in the pyruvate dehydrogenase reaction or pyruvate dehydrogenase enzyme, or it is due to any of the depletion of the coenzyme, the pyruvate will be accumulated. So the pathway of pyruvate is that pyruvate can be converted to lactate. Okay, so both pyruvate and lactate will be present in excess accumulated in the cell, leading to pyruvic acidosis and lactic acidosis. So this is the outcome of pyruvate dehydrogenase enzyme complex deficiency, either due to enzyme deficiency or due to coenzyme deficiency. Is it clear?
Yes, ma'am.
So this deficiency can be inherited one. Mostly it is an inherited defect of the enzyme. It can be a total deficiency of all the enzyme complex, or it can be any one component that is defect. It can lead to pyruvic acidosis and lactic acidosis. And this lactic and pyruvic acidosis will be more prominent when that person have a glucose feeding or carbohydrate diet. When there is a high carbohydrate diet only, this glycolysis will be triggered and pyruvate will be formed, and that pyruvate will be blocked at the level of pyruvate dehydrogenase reaction, and pyruvate will be present in high amount. Some of it will be converted to lactate. So both pyruvate and lactate will be present high in the blood. And due to the presence of pyruvate and lactate, the person will have some neurological symptoms. Okay. And this pyruvate dehydrogenase deficiency will cause lactic acidosis. That will be prominent when seen during hypoxia, when the oxygen level is low, when the patient has circulatory shock, the pulmonary failure, or in diabetes mellitus also, the excess glucose can be channeled to form the lactate. I hope this much is clear for you. By this, we finish up the glycolysis, its regulation, the enzyme, key enzyme, energetics, the link reaction, which is the pyruvate dehydrogenase reaction, its energetics, and the regulation, and also the disorders associated with the enzyme deficiency. Okay, so we'll move on to the next topic, that is gluconeogenesis. Okay, can you see the slide? Gluconeogenesis.
Yes, ma'am. Yes, ma'am.
Okay, so we'll move on to the next topic, gluconeogenesis. As the name indicates, it is neogenesis. Neo means new. So from new substrate, glucose will be generated. That is called gluconeogenesis. The need of gluconeogenesis is that when the blood glucose level is low and the body have less glucose supply, but body needs a certain amount of glucose for the bodily function to continue, especially the RBCs and brain depend upon the glucose for its energy. So blood glucose level should not go down beyond a limit of some 70 milligram per deciliter. Normal fasting blood glucose level 70 to 110 milligram per deciliter. So fasting level should not go beyond 70. If it is below 50, it is difficult to survive. Okay. So when the blood glucose level is getting down and the glucose stores in the body is less, the body will trigger this gluconeogenesis. Means the glucose will be formed from other sources, the sources other than carbohydrate. That is called the gluconeogenesis. So under this gluconeogenesis, we will study the substrates for gluconeogenesis, which means from which all molecules glucose can be generated. Okay, and which are the pathways linking that to glycolysis. We know that only 3 reactions in the glycolysis are irreversible. So if we can, if we have 3 different enzymes which can circumvent all these 3 reactions, the glycolysis can be reversed and it can form glucose from other sources. And we also study about the regulation of the gluconeogenesis, energetics of the gluconeogenesis. So when gluconeogenesis asked us a long answer question, we have to say what is gluconeogenesis, which are the substrates for gluconeogenesis, the reactions, which are the reactions catalyzed, the energy requirement. Actually, it is a energy expensive process compared to glycolysis. More energy is required. Okay, and which are the regulatory factors for the gluconeogenesis and what is the importance? These things you have to study. Okay. Whenever you study about a reaction, you need to know the site of the reaction. What was the site of glycolysis?
Cytoplasm.
So we can say the gluconeogenesis is partly cytoplasmic and partly mitochondrial. So the first reaction is occurring in the mitochondria. The rest of the things are happening in the cytoplasm. Okay. Regarding the definition, it is the process by which glucose molecules are produced from non-carbohydrate precursor. Non-carbohydrate, glucose from non-carbohydrate precursor. Okay. I told you about the substrates, which are the non-carbohydrate precursor from which glucose is formed. These are the 4 non-carbohydrate precursor. First is the lactate or the lactic acid. Lactic acid is not a carbohydrate source. Likewise, glucogenic amino acids, mainly alanine, which is present in the muscle. Other glucogenic amino acid also take, but majority is by the alanine, which is present in the muscle. And in general, the fats cannot be converted back to glucose, but there is an exception. The fat has got a glycerol part. Okay, glycerol part can be converted to fat. How? I will discuss in the coming slide. So glycerol part of the fat is the third thing, and propionate or the propionic acid, that is formed by the oxidation or breakdown of the odd chain fatty acid will produce propionic acid. Even chain fatty acid on degradation will produce acetyl CoA only. Even chain. When there is odd chain, the final one, acetyl CoA, has got 2 carbon only. Propionic acid has got 3 carbon. So when even chain fatty acids are degraded as 2 carbon unit, the thing remaining is all will be 2 carbon acetyl CoA. But in the case of odd chain fatty acid, the last one will be a 3 carbon product, that is propionate or propionic acid. This propionic acid can be channeled to the formation of the glucose. So this is also a gluconeogenic source. The 4 sources, substrates for gluconeogenesis, you should be thorough with. The lactate or lactic acid, glucogenic amino acid, especially the alanine which is present in muscle. Third one is the glycerol part of the fat, and propionic acid or propionate from the or chain fatty acid degradation. That degradation is called beta oxidation. We'll study under the lipid metabolites. Is it clear till now?
Yes.
Okay. So importance is it meets the needs of body when sufficient carbohydrate is not available. I told you in the very first slide, the brain and RBC mainly depend, and RBC solely depend upon the glucose for its energy requirement. And failure of gluconeogenesis will be fatal because it affects both RBC. RBC will be lysed, hemolysis will be there, and the brain function will be deteriorated. So, failure of glucogenesis is fatal to. So when there is hypoglycemia, when blood glucose level falls due to decreased supply of the glucose, the hypoglycemia or decreased blood glucose level will result. That will cause brain dysfunction. Why? The brain depends upon the glucose for its energy, and as a result of brain dysfunction, coma and death can happen. And the other use of gluconeogenesis is that it maintains the intermediates of TCA cycle. You know that oxaloacetic acid, oxaloacetate, it is a 4-carbon compound. It is an intermediate in TCA cycle. That oxaloacetate is formed from the gluconeogenesis. Gluconeogenesis is one of the sources of production of oxaloacetate. And when fatty acids are the main sources of energy, then also oxaloacetate will be formed. Removes excess lactate. I told you when the oxygen supply is low, the glycolysis will proceed to produce the pyruvate, but the pyruvate will be acted upon by lactate dehydrogenase to produce the lactate. That excess lactate can be channeled. That will accumulate in the RBC and muscle. That can be removed by this gluconeogenesis pathway and utilizes the glycerol from the lipid. I told you fat and lipid, they on degradation will produce glycerol and fatty acid. Okay, this glycerol can be utilized to instead of accumulating in the cell or tissue, they can be channeled to form the glucose by gluconeogenesis. These are all the uses of the gluconeogenesis. The purpose. Use propionate from the carbohydrates in ruminants. Ruminants will produce this propionic acid from the carbohydrate. And the humans on degradation of odd chain fatty acid, the final result is last product will be a propionic acid, which is a 3-carbon compound that can also be channeled to the gluconeogenesis pathway. So you can see that it is related to all the 4 substrate. How this all the 4 substrate can be channeled to gluconeogenesis, and that will benefit in the body. Regarding the site, okay, glycolysis occur in almost all cells, and the site or the organelle was the cytoplasm. Here it occurs mainly in the liver. That's why when I told about the Kauri cycle, what was Kauri cycle?
Removal of lactate.
What was it?
Glucose is lactate.
In the muscle,
Muscle
glucose is finally converted to lactate. The lactate itself cannot go back to form glucose within the muscle itself. So lactate comes out of the muscle, enters the blood and reaches the liver. And in the liver, the lactate will be converted to glucose. And this glucose can be easily transported through the blood back to the muscles for its energy need. Okay, so this cycle is the Kauri cycle. So here you can see the lactate will be converted to glucose. So the site is liver only. Okay, mainly occurring in liver. So that thing, Kauri cycle here, the lactate to glucose is the gluconeogenesis. It is occurring mainly in liver and also to some extent in the renal cortex. So you can say the main site of gluconeogenesis is the liver. And the organelle concerned is the mitochondria, partly mitochondria and partly cytoplasm. Some enzyme, one enzyme is occurring in mitochondria, the rest of the enzymes are occurring in the cytoplasm. Okay, and we can call it as a reversal, not completely the reversal of glycolysis. There are some irreversible steps in glycolysis. That steps are overcome by some other steps with some other enzymes. Okay. So lactate transported to liver, I told about the Kauri cycle. Pyruvate to lactate conversion occur when there is an anaerobic glycolysis. That it can be channeled to formation of glucose. Then when there is amino acid, when the glucose supply is in short, amino acids like alanine can be transaminated to form the pyruvate, or aspartate can be. The enzyme is alanine aminotransferase. Likewise, aspartate can be transaminated to form oxaloacetate. That is an intermediate of TCA cycle. So when this amino acid is transaminated, the carbon skeleton formed is a 4-carbon oxaloacetate that can enter the TCA cycle, whereas if it is alanine, it is transaminated to form the pyruvate. Okay, these both can be channeled for the gluconeogenesis. Okay. Oxaloacetate can also finally be converted to pyruvate. Alanine, I told you, which is present in the muscle, is a major amino acid used for the gluconeogenesis. Okay. So the pyruvate to acetyl-CoA, this is the pyruvate dehydrogenase reaction. This is the TCA cycle. In the TCA cycle, you can see this pyruvate can be formed. There are 2 kinds of amino acid in the classification. We have studied glucogenic amino acid, ketogenic amino acid, and both. So glucogenic amino acid can be converted to pyruvate. The major thing we are concerned is the alanine, which is present in the muscle, that can be transaminated to form pyruvate. Okay, and this is the ketogenic amino acid. They can be transferred to the acetoacetate, and also isoleucine can be converted to acetyl-CoA. Okay, these are the glucogenic amino acid. They can also enter the pathway through alpha ketoglutarate. Likewise, some amino acid can enter into the TCA cycle as succinyl-CoA. They are, remember, like valine, isoleucine, methionine, and threonine. They enter as succinyl-CoA. Okay, the details of this amino acid and the entry you will study under the TCA cycle. I'm just explaining an outline only. So alanine can enter into the pyruvate pathway. And one more thing, oxaloacetate I discussed. Aspartate can be transaminated by aspartate aminotransferase to form the oxaloacetate. That can also be used for the gluconeogenesis. This can be used for gluconeogenesis. This can also be used for the gluconeogenesis. Okay, these 2 points you can remember now. Is it clear for you? മനസ്സിലാവുന്നില്ലേ?
Yes, ma'am.
Okay.
Ma'am, previous slide.
Okay.
Thank you.
Okay. So we have discussed about the Kauri cycle. This is the Kauri cycle. Glucose converted to lactate in anaerobic metabolism in the muscle. That accumulation will cause lactic acidosis and fatigue and muscle cramps. That can be relieved by lactate by reaching the blood, finally reaching the liver. The gluconeogenesis happening, finally forming the glucose that can again be transported for the energy purpose of the muscle. This is the Kauri cycle. In addition to Kauri cycle, there is another cycle which is called Cahill cycle. What is Cahill cycle? Is that the pyruvate transaminated to alanine. I told you the reverse is also possible. It is by the transamination reaction. The enzyme is ALT. And that alanine, the pyruvate, why this Corey's and Cahill cycle is happening is because the pyruvate is not permeable from muscle to the blood or to the liver. So that has to be converted to another molecule which is permeable to the membrane, reaching the blood and finally to the liver. That is the alanine which can be transported across the blood to reach the liver. Within the liver, this is a glucogenic amino acid. This is the lactic acid. This is the first substrate for gluconeogenesis I discussed in the initial slide. This is the second substrate for gluconeogenesis I discussed. So alanine can also be channeled to the gluconeogenic pathway, which is in the liver, mitochondria, and the cytoplasm are the two organelles to form the Cahill cycle. Okay, so now you know about the Cahill cycle and Kauri cycle, right? Why are you not responding?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Then now I will tell you about each substrate regarding the Am I audible now?
Yes, ma'am.
Yes, ma'am.
Yes,
Sorry
ma'am.
for the
Yes, ma'am.
connection got low. Okay, so we'll discuss about the next third substrate, that is the glycerol. Glycerol can be channeled to the gluconeogenic pathway. This is by a kinase reaction. This is two-step reaction. The first is by a kinase. Kinase means addition of removal of phosphate. So the phosphate group is added to this carbon. Okay, and for that it requires an ATP. ATP will be converted to ADP. And this formed product is glycerol. CH3OH, CH2OH, CHOH, CH2OH is glycerol. To the glycerol, the third carbon phosphate is attached. That is glycerol 3-phosphate. So you know that by aldolase reaction there is formation of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate form. So glyceraldehyde, instead of glyceraldehyde, here is the glycerol 3-phosphate. That glycerol 3-phosphate can be channeled to this aldolase reaction to form glycerol dihydroxyacetone phosphate by a glycerol 3-phosphate dehydrogenase reaction. Dehydrogenase means removal of hydrogen. So these two hydrogen will be removed and a double bond is created to form the dihydroxyacetone phosphate. Now you can know that this dihydroxy DHAP is an intermediate of glycolysis, so the rest of the reaction can be reversed to form the glucose. Clear? This is how the glycerol part of fat can be channeled to the gluconeogenic pathway. Next is the propionate or the propionic acid. I told you the beta oxidation or the degradation of the fatty acid odd chain fatty acid will yield finally a 3-carbon compound. Propionate, meth, eth, and prop. So 3-carbon acid, COOH. That is CH3, CH2, COH is the propionic acid. To that, if a coenzyme A is attached, it is the propionate CoA. How it can be channeled is that there is a carboxylation reaction. So propionate CoA, addition of carbon dioxide. So one more carbon dioxide can be added. That can be added to here to form this compound is D-methyl. So this is D-carbon. D-methyl malonyl CoA. The rest of the thing is called malonyl CoA. Okay. D-methyl malonyl CoA is formed. It is a biotite. I told you most of the carboxylation reaction or the addition of carbon dioxide, it needs a coenzyme called biotin, which is a B-complex vitamin. So biotin dependent carboxylation produce D-methyl malonyl CoA. You need not worry. This propional coenzyme conversion to D-methyl malonyl CoA and other you'll study under the lipid metabolism. I'm just telling this can also be channeled to the gluconeogenic pathway. How I'm just telling you now, you will get the details when you study the lipid metabolism. Okay, so D-methyl malonyl CoA, D form can be converted to L form. So this COO group is reversed to other side. That is called L-methyl malonyl CoA. And by mutase reaction, it can be converted to a succinyl CoA, which means that the COO can be attached to here to form succinyl CoA. That's 4-carbon compound is called succinyl CoA. You know that this is an intermediate of TCA. Right? That you know, right? Succinyl CoA is an intermediate of.
Yes, ma'am.
Yeah.
So we have discussed about the 4 substrates which can be channeled to the gluconeogenic pathway. Which are those 4 substrates?
Lactate, glycerol,
Lactate,
glycerol,
second is not
propionate.
Second is
Gluconeogenic
gluconeogenic
amino acids.
Which is the amino acid most commonly gluconeogenic?
Alanine.
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 Third is
Glycerol fat.
glycerol part of that. And fourth is
Glycerol.
Propionate
Propionate
from old chain fatty acids.
propionate. Okay. So succinyl CoA can be channeled to the TCA cycle. Okay. And this is the summary of the glycolysis. To say about the gluconeogenesis, we have to first see the glycolysis. These are the steps of the glycolysis, finally forming pyruvate. And this is the first substrate of gluconeogenesis, that is the lactate. So what happens to the lactate is that lactate can enter into the, what happens to the lactate can be reversible by that Kauri's reaction, Kauri cycle, which happens in the liver. Next, about the phase of pyruvate. I told you the pyruvate, which is up to this reaction in the glycolysis, is happening within the cytoplasm, and this pyruvate, there is a pyruvate transporter which is present in the, consider this as a mitochondrial membrane. Mitochondrial membrane has got pyruvate transporters. So pyruvate can easily enter from the cytoplasm to the mitochondria. What is the need of pyruvate entering the mitochondria is that the enzyme for the gluconeogenesis, further conversion of pyruvate, is situated in the mitochondria. The first enzyme which is needed for the gluconeogenesis is the pyruvate carboxylase. Okay. Pyruvate is a 3-carbon compound that has to be converted to 4-carbon compound. That 4-carbon compound is oxaloacetic acid. For that, we need a carboxylation reaction, which means a carbon dioxide is added. I told you most of the carboxylation reactions require which vitamin? As a coenzyme, biotin. Very good. So this is a biotin dependent carboxylation reaction. So carbon dioxide is getting added to the pyruvate within the mitochondria with the help of the pyruvate carboxylase, and it requires 1 ATP also to form the product. Is a 4-carbon compound which is called oxaloacetate or oxaloacetic acid. So this is the first step in the gluconeogenesis. Pyruvate carboxylation reaction. This much happening in the cytoplasm through the transporter. It is entering in the mitochondria. Here, with the help of this enzyme, with this coenzyme, and with 1 ATP, it is forming oxaloacetate, which is the first step in the gluconeogenesis. Okay. So oxaloacetate is formed in the mitochondria, but the rest of the enzyme for the gluconeogenesis is situated in the cytoplasm. So oxaloacetate has to somehow come out of the mitochondria and reach the cytoplasm for rest of the reactions to happen. But unluckily, the oxaloacetate is not permeable to the mitochondrial membrane. So oxaloacetate cannot simply come out of the mitochondrial membrane. Okay. So for the transport of the oxaloacetate along the mitochondrial membrane to reach the cytoplasm, we need a shuttle. That shuttle is called malate aspartate shuttle. Okay, I will tell you. So by pyruvate carboxylase reaction, pyruvate is converted to oxaloacetate by pyruvate carboxylase, which is a biotin dependent carboxylation. What is the fate? Actually, if it is permeable, it will be simple. Oxaloacetate can simply come out of the cytoplasm and rest of the reaction can happen. But unluckily, the oxaloacetate is not permeable. So it has to be converted to some other forms which is permeable to the membrane. Okay. How it happens is that the oxaloacetate by another reaction, MDH is malate dehydrogenase reaction. Malate dehydrogenase. By malate dehydrogenase reaction, it is converted to malate. What is the advantage of malate? Is that malate is able to cross the mitochondrial membrane to reach the cytoplasm, and this reaction can again be converted. This is an interconvertible form, malate to oxaloacetate, but it needs one NAD. And the reversal of this reaction occurs in the cytoplasm, so that at the cytoplasm we got the oxaloacetate. The oxaloacetate is the substrate which we need for the further reactions of the gluconeogenesis to proceed. Clear? Is it clear for you? Do you have class at 10? Do you have class at 10? Please respond.
Recorded class.
Recorded class. Okay. So better you wait for some 15 minutes so that I can finish this gluconeogenesis. Shall I continue?
Yes, ma'am.
Yes, ma'am.
Okay. So oxaloacetate can reach the cytoplasm. And there is one more change that can happen to the oxaloacetate in the mitochondria. I told you about a transamination reaction, aspartate. Aspartate and amino acid can be transaminated to form the oxaloacetate by aspartate aminotransferase. So this oxaloacetate, it is a reversible reaction. So oxaloacetate can be converted to aspartate. Why it is converted to aspartate is that this aspartate is also permeable to the mitochondrial membrane. So aspartate can also reach the cytoplasm, and the reversal of reaction happens. In this way also, oxaloacetate can be regenerated in the cytoplasm. Clear?
Yes, ma'am.
This shuttle is called malate aspartate shuttle. Oxaloacetate itself cannot be traversed through the membrane, so either it can be converted to malate or aspartate to cross the membrane, finally forming oxaloacetate in the cytoplasm. Okay, so this is the first reaction occurring in the mitochondria, forming oxaloacetate. I told you, as it is not permeable by malate aspartate shuttle, it has reached the cytoplasm. In the cytoplasm, which is the second enzyme which convert oxaloacetate to phosphoenol pyruvate is the phosphoenol pyruvate carboxy kinase. You know that the phosphoenol pyruvate and pyruvate all are 3 carbon compounds. But from the pyruvate, we have converted it into a 4 carbon compound, which is oxaloacetate. It has to be reconverted to a 3 carbon compound. So the enzyme is Carboxy Kinase. Carboxy Kinase means 1 carbon dioxide is removed and 1 phosphate is added. The phosphate is added by, it is not ATP, it is GTP. Okay, but both GTP and ATP has got 1 ATP yield. Okay. So here the energy currency is GTP, removal of 1 carbon dioxide. The last carboxylic group was removed, and to the second carbon a phosphate group is attached from the GTP. Okay, so this is called as the second gluconeogenic enzyme, phosphoenol pyruvate carboxy kinase. Is it clear?
Yes,
Okay.
ma'am.
So phosphoenol pyruvate to pyruvate, it was the last irreversible step of the glycolysis. This enzyme was pyruvate kinase. This pyruvate kinase reaction occurs only in single direction. So to reverse it or to circumvent that, we have 2 reactions. Pyruvate cannot be directly, there is no enzyme no one enzyme which converts pyruvate to phosphoenol pyruvate. So this is achieved by a two-step reaction with the help of two enzymes. So first step, as a first step, pyruvate is converted to oxaloacetate, and then this oxaloacetate is converted to phosphoenol pyruvate. So this enzyme was the first enzyme of the gluconeogenesis. So which is this enzyme? First step of the gluconeogenesis, which was the first enzyme? Huh? Are you listening? We discussed about two enzymes of gluconeogenesis. Pyruvate, the 3 carbon converted to 4 carbon with the help of
Pyruvate carboxylase.
it was pyruvate carboxylase, which adds the carbon dioxide with the help of a biotic, and this occurs in mitochondria. This much we discussed, and the product formed is oxaloacetate. It is in the mitochondria, but rest of the enzymes for the gluconeogenesis are seen in the cytoplasm. So oxaloacetate has to reach the cytoplasm. This is achieved with the help of malate aspartate shuttle. Thus, oxaloacetate has reached the cytoplasm. In the cytoplasm, there is a second enzyme for gluconeogenesis, that is from 4 carbon to 3 carbon. This is by which enzyme?
Phosphoenol pyruvate carboxy kinase.
Phosphoenol pyruvate
Carboxy
carboxy
Kinase.
kinase. Here, for that, we require 180. 180p to 180. Here we require 1 GTP to 1 G. Is it clear till now?
Ma'am,
Ah,
and what is this ITP?
ITP is an inosine derivative. Inosine triphosphate.
Okay, ma'am.
We will study that under the nucleotide metabolism. What is inosine? Okay. Nucleotide derivative is the inosine. Guanine is an adenine, guanine, thymine, cytosine. These four are the main nucleotides. As a precursor, there is also an inosine. Inosine monophosphate, inosine triphosphate, all are there. Okay, the details you will study under the nucleotide metabolism. This is not that needed for you. ITP can also be there.
Okay, ma'am.
Okay. So GTP is produced by, from where this GTP comes is that the succinate thiokinase reaction is there in the TCA cycle. Instead of this ATP, succinate thiokinase reaction produces GTP. That GTP is a GTP used here in the phosphoenol pyruvate carboxy kinase reaction. Okay. So pyruvate carboxylase reaction, phosphoenol pyruvate carboxy kinase reaction. By these two reactions, phosphoenol pyruvate is formed, and you know that the other reactions are reversible. So simply the reversal of reaction will happen. Till which step? Till the next regulatory key enzyme, phosphofructokinase. That was not a reversible reaction, right? The others all were reversible reaction. After the first two reactions of key gluconeogenic enzyme, the rest of the pathway is same as the reversal of the glycolysis, and that stops at the step fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate cannot be simply reversed to fructose 6-phosphate. So to circumvent this enzyme, there is another enzyme which is present for the gluconeogenesis. So you can see 1-phosphate is removed. From 1,6-bisphosphate, 6-phosphate is there. Okay. That phosphate removal is by the addition of water and removal of phosphate. What is the name of the enzyme which adds water and removes phosphate called?
8-phosphate.
Adding for removal of phosphate with the help of
Phosphatase.
ATP is called kinase. But removal of phosphate by adding water, that enzyme is called
Phosphate.
phosphatase. Right? We have discussed in the 2,3-BPG shunt pathway about the phosphatase. Adding water to remove phosphate, that kind of enzyme is called phosphatase. So here the enzyme is fructose 1,6-bisphosphatase. It will remove one phosphate from the first carbon so that it will form the fructose 6-phosphate. The next reaction is also reversible. So glucose 6-phosphate is formed. But the first reaction, it is a hexokinase reaction. That was also a key enzyme which was not reversible. Here also one phosphate is removed. It is by addition of water and removal of phosphate. So this is another phosphatase. It is the, which is that phosphatase enzyme? Simply glucose 6-phosphatase. Okay. So can you now tell which are the 4 gluconeogenic enzymes? First is
Pyruvate carboxylase,
pyruvate carboxylase. Second is
pyruvate
Phosphoenol
phosphoenol
pyruvate
pyruvate carboxy kinase,
kinase. Third is
fructose 1,6-bisphosphatase,
fructose 1,6-bisphosphatase. Last is
glucose 6-phosphatase.
glucose 6-phosphatase. Read this today itself, otherwise, and make it thorough. Otherwise, you will forget it. So you have to be thorough about the glycolysis, the enzymes, and the key enzyme. If you know the key enzymes only, you will understand about the key enzymes of gluconeogenesis also. For the pyruvate kinase reaction, the reversal is achieved with the help of 2 enzymes. That 2 are the pyruvate carboxylase and phosphoenol pyruvate carboxylase. These 2 enzymes are for reversal of pyruvate kinase reaction. This is for the reversal of which reaction? Phosphofructokinase reaction. This is for the reversal of hexokinase reaction. Clear?
Yes, ma'am.
Oh. And these enzymes are present in the, I told you the gluconeogenesis, the site is liver and the renal cortex. So this enzyme is present in the liver and kidney. In muscle it is absent. The second key enzyme is also present in liver and kidney, and to some extent with also in the skeletal muscle. So the liver is the organ which can replenish the blood glucose level when the blood glucose level is low due to a low carbohydrate diet. So this gluconeogenesis can provide glucose for some 12 to 18 hours of fasting. So after 12 to 18 hours, the stored glucose which is present in the liver is called glycogen. The glycogen stores will be lost after 12 to 18 hours of fasting. After that, the gluconeogenesis can provide energy, provide glucose to the blood, provide maintain the sufficient blood glucose level for some period. Coming to the energy requirement, you know that the first key enzyme, the pyruvate carboxylase reaction, it requires 1 ATP. Second reaction is phosphoenol pyruvate carboxy kinase. That requires what? ATP or GTP?
GTP.
Okay, 1 GTP. Why it is written as 2? For the synthesis of 1 glucose, 2 pyruvate is needed, right? That is why 2 into 1 ATP. Likewise, here 2 into 1 GTP. Okay. And you know, 3-phosphoglycerate to 1,3-bisphosphoglycerate. That reaction, it produces 1 ATP. Okay. As it is a reversal of reaction, for the reversal to happen also 2 ATP will be required. There. So adding all these 2 plus 2 plus 2, it will give 6 ATP. No energy. We know that hexokinase and phosphofructokinase reaction, it requires 1 ATP. But it is not accounted in the case of gluconeogenesis. Why? Because these enzymes are not there for the gluconeogenesis. Instead of this, there are 2 different enzymes instead of these 2. Okay, so the total energy requirement is 6 ATP. Through glycolysis, we will get 7 ATP or 2 ATP, depend upon aerobic or anaerobic. But it is regarding the gluconeogenesis, it is an energy expensive process. It needs 6 ATP. Clear? Yes or no?
Yes, ma'am.
Yes, ma'am.
Regarding the regulation of gluconeogenesis, it is by induction or repression. Induction or repression, when we say it, it is with the help of hormones. When we discussed about the glycolysis, the main hormone which is involved in, which favors the glycolysis, is the insulin. Okay, you remember, glucose is the hormone which favors the insulin is the hormone which favors the glycolysis. But you know that the gluconeogenesis is just occurring in opposite condition. When glucose level is in plenty, only glycolysis will happen. But when glucose level is in short, it is the gluconeogenesis that happens. So when insulin level is high, it is the repression of the gluconeogenesis. Actually, when insulin is high, it is the glycolysis that is favored. When glycolysis is favored, the gluconeogenesis will be decreased or repressed. In other way, we can call glycolysis and gluconeogenesis are opposite pathway. Okay, when this is favored, this is inhibited. When this is favored, this will be inhibited. So when insulin is present, the gluconeogenesis will be depressed or there will be repression. The hormone which has opposite action of that of the insulin is the glucagon and glucocorticoid. So when they are present, they will induce the gluconeogenesis. Is that clear for you?
Yes, ma'am.
Okay. So how this glucocorticoids favor is that they will induce the transaminases. 2 transaminases I discussed, ALT and AST, alanine aminotransferase and aspartate aminotransferase. So by transamination reaction, we will get substrate, both substrate, oxaloacetate and pyruvate. The end product of this alanine aminotransferase is pyruvate, aspartate is oxaloacetate. Both can be channeled to the gluconeogenesis. So glucocorticoids induce these 2 transaminases enzyme to form the intermediates of glycolysis. Okay. And when there is a high glucagon ratio, it will favor or it will induce the enzymes, key enzymes of the gluconeogenesis, so that the gluconeogenesis is favored. And when gluconeogenesis, the key enzymes of these are the key enzymes of gluconeogenesis. When they are favored, the opposite enzymes, which are the key enzymes of glycolysis, they will be inhibited. Okay, high glucagon will activate the key enzymes of gluconeogenesis and repress or decreases the key enzymes of the glycolysis. Okay, it is achieved by the covalent modification. Covalent modification means the phosphorylation, dephosphorylation reaction. You know that dephosphorylation activates the glycolytic enzyme and pyruvate dehydrogenase enzyme. I told you PDH is active in dephosphorylated form. Likewise, phosphofructokinase is also active in dephosphorylated form. But the enzymes of gluconeogenesis are all opposite to the action of these enzymes. So they will be active in phosphorylated form. So phosphorylated form of this enzyme is the active form. That phosphorylation is favored by these two hormones. Is it clear?
Yes, ma'am.
മനസ്സിലാവുന്നില്ലേ? Why you are not responding?
Ma'am, once more.
Ah. And there is also an allosteric modification. Allosteric regulation you will study under the enzymology. It will be taken by Dr. Meera. An acetyl-CoA, when there is present in high amount, it will activate the pyruvate carboxylase reaction, and also it will inhibit the pyruvate dehydrogenase reaction. Citrate activate. You know that ATP and citrate are the allosteric inhibitors of pyruvate. Which enzyme? Phosphofructokinase. In the case of phosphofructokinase, they are inhibitors. So in the case of gluconeogenesis, it is the opposite. They will activate the fructose 1,6 bisphosphatase. Okay, and AMP and fructose 2,6 bisphosphate, they will inhibit the gluconeogenesis. When you study about the activators, inhibitors, regulators in the glycolysis, you can simply write the gluconeogenesis because it is just the opposite form. Okay. And there is also a reciprocal regulation. Means when one pathway is activated, the another pathway will be inhibited. When glycolysis is favored, the gluconeogenesis will be inhibited, and vice versa. That is called as the reciprocal regulation. And there is a ratio of glucose to urea in the urine. The glucose to urea ratio in the urine, and that ratio will be increased during increased catabolism. Okay, just you need to know that there is a ratio called glucose to urea ratio which is present in the urine. That ratio will be enhanced or increased in catabolic. Catabolic state means when there is starvation, when there is fever, when there is hyperthyroidism, cancer, etc. The body tissues will be broken down. When there is increased broken down of the carbohydrate or mainly protein, that will result in formation of the waste products like urea. The urea level will be increased. Okay. And urea to nitrogen ratio, the urea has 2 nitrogen, right? 2 nitrogen groups will be present in urea. So that ratio is called GN ratio, glucose nitrogen ratio. In animals, the ratio is about 3.65, which means the glucose level is high compared to urea. Okay. So in other words, we can say 1 gram of nitrogen. 1 gram of nitrogen. From where does this nitrogen comes? From the protein in the body which gets degraded. That will generate some 3.65 gram of glucose. Okay. Just need to know that there is a term called glucose nitrogen ratio. The normal value is 3.65. I hope this much is clear for you. So we have discussed about the gluconeogenesis, the key enzyme, the regulation, the energy need for that, the substrates, how the substrates can be channeled through the gluconeogenic pathway, how the irreversible steps are circumvented from the glycolysis to gluconeogenesis, about the regulation in details. Is it clear till now? Yes or no?
Yes, ma'am.
Okay. So we'll continue
Yes,
on
ma'am.
Thursday. Read thoroughly and come. I will randomly call 2 or 3 of you to tell the summary. If you don't tell the summary, I will mark you as absent.
Ma'am,
Ah,
ma'am, can you please upload this PPT in Google Classroom?
no. If you want, you have to take down the notes. I won't give you the PPTs. ഞാൻ ആദ്യമേ പറഞ്ഞിട്ടുള്ളതാണല്ലോ ഇത്ര ടൈം സ്പെൻഡ് ചെയ്ത് ഓരോ സ്ലൈഡിനും അത്രയും ടൈം തന്നിട്ട് തന്നെയാണ് എടുത്തിട്ടുള്ളത് സോ ഈ സ്ലൈഡ് തരാൻ പറ്റില്ല സോറി ഓക്കെ താങ്ക് യു
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
താങ്ക് യു മാം